Interaction of the maxillofacial region organs with various body systems, respiratory and communication functions. Practical work number 1. Determination of the functional mobility of the tongue taste buds before and after meals. Functional mobility of receptors is understood as a phenomenon that is expressed in the weakening or strengthening of the activity of analyzer system by reducing or increasing the number of active receptors. The level of mobilization of taste buds mainly depends on the motivation of hunger or the taste of satiety. Aim of the work: master the method of studying the functional mobility of the taste buds of the tongue and make sure that food intake leads to the mobilization of taste buds. Required for work: glass capillary with a curved tip, the diameter of which approximately corresponds to the size of the fungiform papilla. Glass, tray, filter paper, sand glass 1 minute, 45% of sugar solution, transparent and tint with food coloring or magenta. Work sequence: the study is carried out on students on an empty stomach or at least 3 or 4 hours after the last meal. The tongue is dried with filter paper. Colorless taste stimulus is applied to individual tongue fungiform papilla using a capillary. At the same time, 4 papilla are identified which give a sensation of sweet taste, and they are marked with a tint stimulus. This constitutes one sample. In total, 5 samples are carried out in the experiment with an interval between them of 1–2 minutes. After each test, the mouth is rinsed with water. Examine the same papilla. The appearance of a taste sensation is marked with a plus sign. The absence is marked with a minus sign. The total number of positive responses is calculated and the level of mobilization is expressed as a percentage. The study is repeated after a meal, a glass of sweet tea with cookie. Formulation of the protocol. Enter the results of the study into tables. To draw a conclusion, compare the level of mobilization of the taste buds of the tongue before and after eating to indicate the mechanism of interaction of taste and visceral analyzers. Before eating, all the taste buds were mobilized. After eating, a signal was sent to the central nervous system. The taste buds were demobilized because their sensitivity decreased. Practical work number 2. Determination of the role of the olfactory analyzer in the occurrence of taste sensations. Aim of the work: make sure that the olfactory analyzer is involved in the emergence of a sense of taste. Required for work: onion or candy with durian extract. Also, you can use nose clip. The study is carried out on a person. The researcher chose a piece of candy. First with the nasal passage opened and then with the closed ones, and compares the sensations. Formulation of the protocol. Describe the results of the study. With the participation of the olfactory analyzer, the taste of food is felt, and in absence, the food becomes tasteless. To draw a conclusion indicating why the sense of taste is partially lost with a runny nose. With a runny nose, a person begins to breathe more often through the mouth, which causes dry mouth and chemical irritants in food to dissolve less easily. Practical work number 3. Pneumography at rest, when reading aloud, when chewing and swallowing. Pneumography is a method of recording the respiratory movements of the chest, which allows you to determine the duration of inhalation and exhalation, the frequency and depth of breathing. The nature of a pneumogram is different with calm and with speech breathing, as well as during digestive function. Aim of the work: master the technique of pneumography. Compare the pneumogram obtained with calm and speech breathing, chewing and swallowing. It's performed in the functional diagnostic training room. Required for work: pneumograph which has respiratory and temperature transducers. Work sequence. Turn on the computer. Turn equipment. Respiratory transducer to channel number 1. Temperature transducer to channel number 2. Wrap the breathing transducer around the chest below the armpits and above the nipples. Attach the respiratory transducer to the patient's face. Start work in a biopack student's lab. Choose the lesson number 8 and start the recording. A pneumogram is recorded when breathing calmly, when reading aloud, when chewing cookies, when swallowing. Look at the graphic and mark calm breathing, speech breathing, breathing during chewing, and breathing during swallowing. Formulation of the protocol. The results of the analysis of the pneumogram should be entered to the table. Draw a conclusion by indicating the differences between the pneumogram of speech breathing and the pneumogram of calm breathing, in terms of the depth and duration of inhalation and exhalation. With calm breathing, involuntary inhalation and exhalation are the same in duration, and during speech breathing, a deeper inhalation and a slow exhalation. Also draw a conclusion by indicating features of breathing during chewing and swallowing. During chewing, the inhale is deep, followed by rapid breathing, while swallowing there is a breath delay. Conclusion. In this video, we found out that the process of taste formation largely depends on interaction with other body systems, and also that the organs of the maxillofacial region play an important role in the breathing process.